Hey team, Jack here. So today I'm just going to jump into the cytotoxic T cell, and it's actually quite a simple cell compared to the T helper cell. So let me just jump into it. Here we have the T cell receptor. Now remember, in a cytotoxic T cell, you have the CD8 molecule, and the CD8 molecule recognizes MHC1. And MHC1 is present on all cells in your body except your red blood cells and platelet cells. And what it does is it samples an antigen from inside the cytosol of the cell and displays it, right? And a cytotoxic T cell will recognize it if it's only if it has the particular antigen that cytotoxic T cell is designed to recognize. So each cytotoxic T cell in your body is designed to only recognize one antigen, and that antigen will be different, will not be a self-antigen, because we have a selection process during the maturation of T cells that will kill off any T cells that recognize a self-antigen. So the T cells in your body will ideally only recognize a pathogenic antigen, right? So a good example would be a cell infected with a virus. Will sample some of that viral protein, display it on its MHC1 molecule, and perhaps this one T cell receptor, which is completely unique to that T cell because it self-genetically engineered itself to have a completely unique T cell receptor, will recognize that antigen and then be activated. Now, when I want to talk about a cytotoxic T cell, I always want to talk about natural killer cells, which I've already covered in a previous video. Natural killer cells are an innate immune cell that contains granules, and they too bind to the MHC1 molecule. So here we have a lung epithelial cell with a normal protein and a fragment of that normal protein displayed on the MHC1 molecule. The natural killer cell will come and bind to it, and it will recognize it, because a natural killer cell is designed to recognize self-antigens. So it will recognize it, and if it does recognize it, it will not kill the cell. However, if that lung epithelia is affected with a virus like SARS-CoV-2, it will display a viral protein on its MHC1 molecule, and that will not be recognized by the natural killer cell, and the failure to recognize a self-antigen results in its activation. So a natural killer cell, if it cannot recognize the antigen displayed on the MHC1 molecule, it will activate. And its activation results in degranulation, and in those granules is a protein called perforin, and perforin forms pores in the membrane. And by forming pores in the membrane, you essentially kill and lyse that cell, right? You tell that cell it needs to die. The cell can undergo apoptosis, or if it's bad enough and it's not functioning, those pores will just eventually kill that cell. There's also other compounds in the granules like gran enzymes that will go in and activate certain cell death pathways. So they will be released from the granules and they'll flow in this pore into the cell and activate cell death pathways. So gran enzymes do that from inside the natural killer cell. But the cytotoxic T cell is basically the adaptive immune system's version of the natural killer cell. However, it's got a couple of distinct differences. And with the natural killer cell, it's the failure to recognize a self-antigen that results in activation and killing of that cell. With a cytotoxic T cell, it is the recognition of a pathogenic antigen. So in the case of a TC cell, the receptor needs to recognize and activate a foreign particle. So it's the recognition in a cytotoxic T cell that results in its activation. But once it's activated, it pretty much does the same thing. It produces perforin and gran enzymes that create a hole in the membrane and initiate cell death pathways in that lung epithelial cell. It could be any cell in your body. That's just an example of a lung epithelial cell. The one slight difference is natural killer cells store those proteins in granules, which allow for very fast release, whereas cytotoxic T cell actually just has a bunch of rough endoplasmic reticulum to pump out that protein. So it pumps out that protein through traditional secretion pathways, right? through the rough endoplasmic reticulum. But it still produces perforin and grain enzymes to initiate cell death pathways in the cell. So the cytotoxic T cell is really just the natural killer cell of the adaptive immune system. But like how the innate immune system is non-specific, right? The natural killer cell is part of the innate immune system. It's non-specific. And how does it do that? Well, it's saying, you don't look like me, therefore I'll kill you, right? So that's non-specific. I don't know what pathogen you're showing me, says the natural killer cell. I just know you're not showing me a self-antigen, so I will kill you. Whereas the T cell, cytotoxic T cell, is a part of the adaptive immune system, which is a targeted immune system, and it says, I recognize that antigen as a pathogen. I know you are showing me a disease, therefore I will kill you, right? So there's that subtle difference there between the natural killer cell and the cytotoxic T cell. But also activation will result in the division of this cytotoxic T cell, and the cytotoxic T cell will go on to create a whole bunch of memory cytotoxic T cells, which are essentially just the exact same version of this cytotoxic T cell. Now remember, because this cytotoxic T cell has genetically modified its own self to create a T cell receptor that will only recognize the antigen that was displayed by that MHC1 molecule. So this viral protein, for example, here, this could be a spike protein from COVID-19. By dividing and dividing and dividing, those cells that it divides, those clonal cells, will have those same genetic mutations that it did to itself. It has the same unique genome specific to that T cell that's different to every other cell in your body, just in a few genes. But we're told that every cell contains the same recipe book, but that's not true with adaptive immune cells. They get to rewrite a few of their pages. And so we need to create more of those cells in case we get infected with that disease again. So the cytotoxic T cell will proliferate, and each will have that unique genome that can recognize that spike protein with the T cell receptor. And we'll end up with loads of memory cells, but we'll also end up with effector cells. So the effector cells are the ones that are going to go out and kill more cells that display this viral protein. Whereas the memory cells won't go do that. They'll just go sit and wait to be activated in the next infection. Where do those memory cells go? Well, they don't just go to the lymph node. They actually go everywhere. And that's because they need to be ready for when the disease comes again. So sites of, they go throughout the body, and you will find them in the lung parenchyma, which is the tissue of the lungs, the airways of the lungs, and the lymph nodes, and the spleen, and the liver, and the peritoneal cavity. You will find them in the bone marrow. You'll find them all throughout your body. These memory T cells are ready to respond. They are there, ready for the next infection. And obviously you can start to see, I'll do a video just on this, but you can start to see how vaccines work, right? Vaccines have caused the proliferation of cytotoxic T cells, but also T helper memory cells, and also B memory cells. So now our immune system has all these cells that are there and have genetically engineered themselves to be the perfect cells to respond to that pathogen should it come again, right? So yeah, it's brilliant. So that's, roughly how vaccines work, but I'm going to go into a lot more detail in a later video on vaccines. And up next, in our next video, I'm going to cover cytokine bead assays, because I always like to chuck a few method videos in there. So if you ever wanted to go to the literature and read actual scientific research, you understand what's going on because you understand the methods that they've used.